Welcome back. In the last web lecture, we looked at some general principles of movement of materials through plants, and we focused mostly on short distance transport. So in this web lecture, we're going to start to look at the long distance, the bulk transport of water and solutes throughout the plant, and this is going to be happening in the xylem. So remember that we said diffusion, active transport, and bulk flow act together to transport resources throughout the plant. And so if we look at the bulk transport part of this, the transport through all of these vessels throughout the plant that we can see here in the leaf, we're going to see that all of these mechanisms play a role in the bulk transport of water and solutes throughout the plant. And it's going to do this through a process called transpiration. So transpiration we've seen in very general terms before when we're thinking about water loss through stomata and adaptations in, for example, C4 and CAM plants. And so we saw that water loss is an inevitable consequence of a plant's need to obtain carbon dioxide for photosynthesis. If you are able to take things in, you can also have things leaking out, and we end up losing quite a bit of water through these openings. So evaporation from leaves offers both challenges and benefits to plants. So evaporation brings a flow of water, carrying minerals from roots to shoots and cooling the plant. However, if that water is not replaced, plant cells will dry out and die. So we need to have this constant source of water to replace the water that's lost to keep all of this transport moving. So the process of transpiration results in water transport from roots to shoots due to differences in water potential, and it's going to be driven by the evaporation of water from the stomata of leaves. So transpiration occurs whenever two conditions are met. The stomata have to be open to be able to evaporate that water out from the leaves, and the air surrounding the leaves has to be drier than the air inside the leaves to draw that water out. So let's sort of start at the root side of this and see how this water and these minerals are taken in by the root cells. So most water and mineral absorption occurs near the root tips, where root hairs are located, and the epidermis is very permeable to water. Root hairs, remember, are these long, fine extensions of the epidermal cells, and they elaborate the surface area of these epidermal cells enormously. So root hairs account for much of the surface area of these roots. After the soil solution enters the roots, the extensive surface area of cortical cell membranes enhances the uptake of water. So now this water is part of the environment of the cortical cells underneath the epidermis. And so now all these cortical cells are going to participate in the uptake of water also, with all of their surface area. Active transport, using ATP to actively move molecules into the cell, enables essential minerals to accumulate at much higher concentrations in roots compared to the surrounding soil. So we're going to concentrate these minerals within the roots. So remember that the endodermis is the innermost layer of cells in the root cortex. So it surrounds the vascular cylinder and is that last checkpoint. And it's going to be a selective barrier that's going to allow needed materials in and keep unwanted materials out of the vascular tissue. So if you remember from the last web lecture, those symplastic and apoplastic pathways through all of these layers of cells, water can cross the cortex via either the symplast or the apoplast. So if you follow these lines, the pink lines indicate the apoplastic route going around the cells through the cell walls and through the intercellular spaces. The symplastic route is going to enter the cell and travel within the cells through the plasmodesmata. Once they get to the endodermis surrounding the vascular cylinder, there's going to be a waxy material surrounding this endodermis called the Casparian strip. So the Casparian strip is impermeable to water or minerals, so everything is completely blocked at that point. So water and minerals that are in the apoplast that are outside of the cells have to cross into the endodermis cells across the cell membrane to be able to enter the vascular cylinder. So at some point, every molecule that enters the vascular cylinder has to cross the cell membrane and go to the interior of a cell to use that selectively permeable membrane of the cell to determine whether material should be allowed to cross or not. So basically every water molecule and every mineral molecule has to at some point cross a cell membrane to get access to the vascular cylinder. If they're going through the symplastic route, they have to cross a cell membrane to get into the interior of a cell to begin with, and then they've already been checked. They can go right on through the plasmodesmata all the way through into the xylem. If they're coming in through the apoplastic route when they get to that Casparian strip, then if they're going to go any further, it has to be through the cell membrane of one of these endodermal cells to be able to gain admittance into the vascular cylinder. So the endodermis regulates and transports needed materials from the soil into the xylem. And remember that the cells of the xylem, the tracheids and the vessel elements, are non-living tissue. They're part of the apoplast. So they can't take up this material. The endodermal cells are going to discharge the water and minerals from their protoplasts into their own cell walls. So now they're back into the apoplast, and they can travel from this apoplast into the non-living cells in the vascular cylinder. Diffusion and active transport are involved in this movement from the symplast to the apoplast, so they have to be actively discharged from these endodermal cells. Once in the apoplast, water and minerals can enter the tracheids and vessel elements by diffusion and enter the vascular system. So the question is whether this movement of water and solutes through the plant is due to pushing from the roots, or are they pulled through by the leaves? And the answer is a little bit of both. So let's first take a look at root pressure. So remember that the cells of the root are actively transporting minerals from the soil into the root cells and accumulating, creating a higher concentration of minerals within the cells than outside in the soil. So at night, root cells continue pumping mineral ions into the xylem of the vascular cylinder, and this is going to lower the water potential by increasing the concentration of solutes. So this is going to cause water to flow in from the root cortex, and this is going to generate a root pressure, a push of the xylem sap. So the xylem sap refers to the water and everything that's dissolved in it. So root pressure sometimes results in a phenomenon called guttation, the exudation of water droplets on the tips or edges of leaves. Remember, do not get this confused with dew. Dew is just condensation from the moisture that's in the air onto the surface of leaves. This is actually coming out of the vascular system due to that root pressure during the night. So this positive root pressure is happening all the time due to the accumulation of minerals by active transport into the root cells, but this is a relatively weak pressure, and it's going to be opposed by gravity, and so before it gets very far up into the plant, it's going to be balanced by the force of gravity, and it's not going to be able to transport that water a very long distance from the roots. So it's clear that this is not the whole story of how the water moves through the plant. So now let's look at this from the other end, pulling the water up from the leaves, and this is known as the cohesion-tension hypothesis. So according to the cohesion-tension hypothesis, transpiration, this evaporation of water from the surface of the leaves, and water cohesion, so remember this is one of those emergent properties of water due to hydrogen bonding, the attraction between the partial negative charge on oxygen and the partial positive charge on the hydrogen parts of water molecules that tends to make them stick together. So this combination of transpiration and the cohesion of the water molecules is going to start a pull up at the shoots, and that pull is going to be transmitted all the way down to the roots. Xylem sap is normally under negative pressure or tension, so a pulling pressure. So just to review quickly this idea of water potential, because this is when we're going to start to use this concept, it's going to become very important. So keep in mind, the important things to remember is that water flows from an area of higher water potential to an area of lower water potential. And so in this case where we have a flaccid cell, that's put into a more concentrated solution, we know just intuitively from understanding osmosis that the water is going to flow out of that cell to the higher solute concentration. And we now know that this is because there's a higher water potential inside the cell than outside the cell. This negative 0.9 megapascals is lower than the negative 0.7 megapascals inside the cell, and water is going to flow from the higher water potential to the lower water potential. Then if we take into account pressure potential, if we take this flaccid cell and put it in pure water so that we have a greater water potential outside the cell than inside the cell, water is going to flow into the cell and it's going to start to create turgor pressure that's going to create a pressure potential, and we know that water will stop its net inward flow. Remember, it's going to reach an equilibrium, which means water is just as likely to flow in as flow out when that pressure potential is the same magnitude as the negative solute potential. So there's this balance between pressure and solute potential that's going to cause an equilibrium. So now let's think about the water potential in air. This is kind of our starting point where we're going to start to create this pull on the water column within the plant. So in the atmosphere, water exists as a vapor with no solutes in it, just pure water in the atmosphere. So it's going to have a solute potential of 0 megapascals. But there's going to be a pressure potential associated with the water vapor, and it's going to depend on temperature and humidity. So when air is dry, there are relatively few water molecules, and because of that, it's going to exert a relatively low pressure potential. When the air is warm, water molecules are moving a lot, they're going to tend to be farther apart, and also exert lower pressure. So warm, dry air has a very low water potential, and evaporation happens most easily under these conditions. So in most cases, the water potential is highest in the soil, lower in the roots, lower still in the leaves, and the very lowest in the atmosphere. So what this does is it creates a water potential gradient going from high in the ground all the way up to the very lowest in the air. And so to move up a plant, the water moves down the water potential gradient from high to low between the soil, its tissues, and the atmosphere. So when it does so, it's going to replace the water lost through this evaporation into the atmosphere. So let's take a look at how this water is replaced when it evaporates off of the leaves. So water vapor in the air spaces of a leaf diffuses down its water potential gradient. Remember, it's very low water potential out here in the air. Here's a stoma, and the water vapor is diffusing away from these air spaces. So now if we look into a close-up into the outer cell wall of one of these mesophyll cells, so this is what we're looking at here, a little bit of the cell wall, and we're going to look at a close-up of these microfibrils within the cell wall and the spaces in between them. We can see here the water vapor is evaporating off and the water is going to be replaced from within the cell. So as more and more water is pulled off by evaporation, this air-water interface is going to retreat back. The water's coming off, and the surface tension is going to cause this little meniscus to form that's going to have a great deal of tension associated with it as those water molecules are trying to stay together under this negative pressure. So increased surface tension pulls the water from cells and air spaces. So the surface tension of water at the air-water interface creates this negative pressure potential. So that negative pressure potential is going to lower the water potential. And with this decrease in water potential, water is going to move from someplace with a greater water potential. So water molecules are going to be pulled from more hydrated areas of the leaf by this negative pressure potential created at the air-water interface within the cell wall of this mesophyll cell. So then the cohesion of water molecules transfers those pulling forces to the water in the xylem. So as this air in the air spaces becomes drier, as water is moved due to this evaporation, the cohesion of water molecules is going to start pulling water out of the xylem and into these air spaces and into the cells that are losing water. And this transpirational pull on the xylem sap is going to be transmitted from the leaves all the way to the roots by the cohesion of the water molecules. So we're going to have an uninterrupted, constant column of water molecules, unbroken, all of the water molecules sticking to each other and being pulled up by this negative pressure at the top. So the water molecules exiting the xylem tug on the adjacent water molecules just below them, down the column. And the adhesion of water molecules, so this tendency of the water molecules to form hydrogen bonds with the hydrophilic molecules in the cell wall, is going to make them stick to the walls of the xylem a little bit, and that's going to help to resist the downward force of gravity. So we can think of this negative pressure just exactly like what happens if you suck water up through a straw. You're exerting a negative pressure with your mouth, and it's the cohesion of water molecules that's keeping the water flowing up into your mouth. If you've ever tried to suck up water through a straw that has a break in it so that air is getting in, you know that you lose that negative pressure and the straw doesn't work nearly as effectively because you've lost the cohesion in the water molecules because of the intrusion of air. So we need some thick secondary walls to prevent the vessels from collapsing under that negative pressure. And so just like you need a little bit of stiffness in your straw to be able to use it, or if you think of a vacuum hose that has these reinforcing rings around it to keep it from just collapsing under that negative pressure. But there are situations that can cause a little bubble to form and disrupt this continuous column of water molecules. So drought conditions or freezing can cause a phenomenon called cavitation, where you get a little bubble of water vapor within the water column. And this is going to disrupt that column of water pulling on each other and keep the water from moving through that vessel. So it turns out that living plants have some ways to get around these cavitation bubbles. So xylem sap transport can often continue after these cavitation events by either moving between adjacent xylem cells through the pits. So remember, the tracheids have these little pits along their length. If the water leaves through those pits and goes into an adjacent tracheid, it can continue moving up and around that cavitation bubble. There's also some recent evidence that suggests that xylem sap can move between xylem and phloem to be able to circumvent these cavitation bubbles. So in some cases, cavitation can be repaired by various mechanisms. And then, of course, every year, new xylem is added by secondary growth. So if you lose some vessels because you get a bubble in them and they're not functioning, they weren't going to be functioning forever anyway, this new growth will take over that function of water transport and the plant will be fine. So let's just review what we know about this transpiration cohesion hypothesis. So the movement of xylem sap against gravity is maintained by the cohesion-tension mechanism. So it does not require any energy on the part of the plant. So bulk flow is driven by a water potential difference at opposite ends of the xylem tissue, from the maximum water potential down in the soil to the minimum water potential up in the atmosphere. That's going to drive the movement of the water from high water potential to low water potential. So bulk flow is driven by transpiration and does not require energy from the plant. Like photosynthesis, it's completely solar powered. So the warmth from the sun is going to encourage this evaporation from the leaves and power this whole process. So bulk flow differs from diffusion in several important ways. So it's driven by differences in pressure potential, not in solute potential. So remember, diffusion is driven by concentration gradient, differences in solute concentration in one area compared to another. This is more driven by the pressure component of the water potential. It occurs in hollow dead cells, not across the membranes of living cells, so it's a little bit different from osmosis in that respect. There's not that semi-permeable barrier that we think of when it comes to osmosis. And it's going to move the entire solution, both the water and the solutes. Unlike osmosis, in which just the water moves, or diffusion, in which just the solutes move, it's going to move everything together. And it's much faster than simple diffusion. Because it's being powered by these differences in pressure potential, you can get a lot more force behind it to increase the rate of transport. So what does determine the rate of transport of water in the xylem? So it's going to depend on the rate of transpiration, which in turn is going to be regulated by these little openings called stomata that we've been hearing so much about. So remember that leaves generally have large surface areas and high surface to volume ratios. And these characteristics are going to increase the rate of photosynthesis, but also will increase the rate of water loss through the stomata. So remember that stomata have these little guard cells flanking them that are going to open and close to help maintain water balance at desired levels, and it's going to help to balance water conservation with the need for gas exchange for photosynthesis. So let's take a closer look at how this happens. So the opening and closing of these stomata is going to be determined by changes in turgor pressure, and that's going to open and close these stomata. So remember, turgor pressure is the pressure of the increasing volume of the cell contents as water enters through osmosis, and then that cell wall pushing back against that. So remember, this is a positive pressure. When these guard cells are turgid, they're going to tend to bow outward, and then that's going to cause the pore between them to open. So they're going to have this change of shape due to that turgor pressure that's going to open up that hole. When those cells are flaccid, the guard cells are going to lose that bowed shape and come together, and that pore will close. So what causes these changes in turgor pressure? So they are the result primarily of the reversible uptake and loss of potassium ions by the guard cells. And so this is going to occur due to an electrochemical gradient that's produced by the proton pumps, the hydrogen ion pumps. So the hydrogen ion pumps create a relatively negative charge inside, positive charge outside, so that when those potassium channels open up, the positively charged potassium ions flow into the cells, creating a high concentration of potassium inside. Water is going to follow by osmosis because that's going to increase the solute potential inside those cells, causing those cells to become turgid. When that voltage is balanced, potassium ions are going to tend to move out of the cell along their concentration gradient, and again, water will follow by osmosis, and then these cells will become flaccid, closing off the stomata. So what causes these stomata to open and close? So generally, stomata open during the day and close at night to minimize water loss. This is the case in C3 plants. And the opening of the stomata in the morning is going to be triggered by light, also the depletion of carbon dioxide, and also there's going to be an internal clock that's going to regulate the opening and closing in these guard cells. So all eukaryotic organisms have these internal clocks that create what's called a circadian rhythm. And so these are 24-hour cycles of different activities that occur in biological systems. So various things can cause stomata to close during the daytime. One of them is drought stress, when the plant becomes severely water stressed. Even though they need to have them open for photosynthesis, the water loss is more important at that point. There's also a hormone called abscisic acid, ABA, that is produced in response to water deficiency and causes the stomata to close. So transpiration can have negative effects as well on plants. So sunny, warm, dry, and windy conditions cause a high rate of evaporation and an increase in these transpiration rates. So even with the stomata closed, there's going to be some evaporative water loss across the cuticle, a very limited amount, but that will happen also. And then if the uptake of water and the transport of the water are not sufficient to replace whatever water is lost through the cuticle, the plant will wilt, and this is what we know of as the sort of droopy looking wilted plant. Transpiration is also very important in thermal regulation in plants. So plants can also overheat. Overheating causes proteins to denature, and that evaporative cooling that happens during transpiration is going to be effective in lowering the temperature of the leaves. So in this web lecture, we've seen how this whole process works of moving water all the way from the roots up to the very tops and the leaves in the very tallest of trees. So it all begins at the roots with the uptake of water and minerals from the roots, and they're pushed into the roots under positive pressure due to the active uptake of minerals. The water and solutes that have entered the roots are going to be pulled up to the leaves by this process of transpiration and cohesion/adhesion of water molecules. That's going to be the main pulling force that's going to allow this water to move up very great heights against the force of gravity. And then finally we saw that stomata are the main mechanism for regulating the rate of transpiration and managing that balance between the need for water and to minimize water loss and the need for gas exchange for photosynthesis. So in the next web lecture, we're going to take a look at the other side of this, the distribution of sugars that are produced in the leaves through the phloem and throughout all the tissues in the plant that require them for energy.